Hello and good day. My name is David John, and I'm going to be covering part of the microbiology modules for the Brewing Arts program. I have a position currently as a professor in the University of South Florida, St. Petersburg, where I teach microbiology and genetics. And I also am the professor who manages the internship program that we have with several of the breweries, the craft breweries in St. Petersburg and Tampa. So the current module that we're going to be working on right now is for microbiology, and the components that I'm going to be covering are microbiology basics, which is the module that we have right now, and also the metabolism of wort by yeast module I will also be instructing to you. So what we're going to look at in this module covers some of the basics of what microorganisms are, including mostly yeast as well as bacteria. So to do that, we need to first consider what life is, what microorganisms are, what sets them apart from other things that are not living. But we also really need to get into some of the chemistry that makes life work on this scale especially. So we're going to be covering some of the basics of biochemistry, as well as then the anatomy and the workings of cells, such as the yeast biology and structure. We'll look a little bit more closely at yeast biology, and then also we'll cover some aspects of bacterial biology. So this is a very condensed kind of content that I'm going to be presenting that is mostly going to focus on just the basics. So it's not going to be the same level that we would go through in a semester-long course, naturally, since we're just doing this one lecture. But I think we'll get a lot of the important basic ideas out there for you. So we'll now move on to parts of the lecture. So as we move into this material, I am going to be just showing the lecture slides rather than the picture of my face, because this is, of course, what's more important to you. So as we begin, we'll first talk a bit about what is life and how some important work by our father of microbiology and one of the most important people in the understanding of brewing worked on this, and that, of course, is Louis Pasteur. So first we want to think about what is it that sets apart a living thing or an organism from something that would be considered non-living. And there are several attributes that we can identify, but three of the most important ones that are shared and critical to the definition of anything that's alive is that it's based on cells. So all organisms that we know of on Earth have their basic construction is that they're made of what we call cells. Another aspect that defines a living organism is that it's able to reproduce itself. If it wasn't able to reproduce, then it wouldn't be around anymore. It would be extinct. And then also that it can perform metabolism. And so what that means is essentially that it can take in some forms of energy from its surroundings, either chemical energy or light energy, and make a conversion of that to the kinds of energy, chemical energy, that can be used within the cell. So much of our current understanding and appreciation of how cells do compose living organisms, as well as the importance of living organisms such as yeast in the production of beer, has come from Louis Pasteur. Louis Pasteur is French, or was a French chemist and microbiologist who lived and worked in the 1800s. So one of the key and really significant accomplishments and achievements that he is responsible for is demonstrating that the fermentation that occurs to produce beer and wine is actually due to living organisms. There was competing ideas during that time that the fermentation reaction was simply something that was chemical, and the living cells that developed in beer were somehow just a byproduct of that, or were not directly related. Pasteur ultimately showed that it is, in fact, the yeast that live or that exist in the beer that is what produces the components that we value for that, which is namely the alcohol as well as the carbon dioxide. Another important achievement of his is that he was able to show that and confirm really that all cells or all living organisms have to come from pre-existing cells or from other organisms. This was in contradiction to the hypothesis of spontaneous generation, that whenever living cells arose from substances that didn't appear to have them, that it was actually due to the introduction of a small maybe a small number of those cells that began to proliferate. And so this is the kind of the crux of the idea that all cells come from pre-existing organisms, and it's in contradiction to the idea of spontaneous generation. Spontaneous generation would have been that just due to the nutrients present in some piece of meat or nutrient broth or grape juice, that the living organisms were able to assemble within that and come to life. And so his work to disprove this, spontaneous generation, is what essentially makes up one of the main components of modern cell theory. We should be a little bit familiar with the nature of his key experiment and how that came to disprove spontaneous generation. And this is a very famous classic experiment. And the way it was done is that he created these glass flasks that were called swan neck flasks. And you can see an idea of what a swan neck is, where there's a long extension of the glass as a tube on top of this bulb. And he could create or introduce nutrient broth into this flask. And then by heating it and boiling, he would essentially successfully destroy all the living cells that were in there. And so if we can follow the path of this experiment, if one were to heat that broth and sterilize it adequately and let it sit, even though it's open to the air, as far as there being gas exchange being able to go in, because of the shape of the flask, no particles could get into where the broth was. And so in those cases, there would not be any development of living cells or bacteria to be found in there. However, if one were to do that same process and then break the swan neck off, that would allow particles to come in from the air, like spores of mold or bacteria that might be carried on dust, then after some time, bacteria would develop. Likewise, if one would apply the heat and then tip the flask a little bit to the point in the swan neck where some of those particles could have been settled, and then tip it back and you could get bacteria. So these steps basically were his classic famous experiment that disproved spontaneous generation and showed that you need to have some particles that would carry cells introduced in order to obtain cells. And this is significant for us today in terms of our understanding of how we need to maintain sanitary conditions and what we need to do to prevent unwanted contaminating organisms from getting into our products, our food products, such as, of course, beer. Another one of his major accomplishments then was the practice of pasteurization. So Pasteur, in conducting these experiments and doing further work, also showed that heating can kill the bacteria and fungi that are responsible, the pre-existing cells that are responsible for spoilage of a lot of different foods. So he mostly was working on milk and beer, as well as wine. And so at the time, he observed that beer didn't last too long. And so by doing experiments and showing that by heating that beer after fermented, that once it was packaged, it stayed good for much longer. And so that went along with basically discovering that the bacterium that can get into these drinks or these beverages can proliferate and produce acids that can cause that souring or spoilage. The types of organisms that can do this, we recognize now, are mostly lactic acid bacteria or acetic acid bacteria. And so that the names of those acids, lactic or acetic, is describing what they produce. So lactic acid bacteria produce lactic acid. And we'll look at some examples of those kinds of bacteria later on in this unit. So he discovered and demonstrated that heating of the beverage to up to 60 to 100 degrees Celsius, which is about 140 to 212 degrees Fahrenheit, 212 degrees is the boiling point, will kill the bacteria, and that will prolong the quality, the stability of that beverage once it's been packaged. So that's where pasteurization comes from, and that's what it is. Basically just heating your product, your beverage, to kill the cells that are in it. So this led to the components of the modern cell theory, that all organisms are made of cells, and that all cells in existence have to come from pre-existing cells, rather than from spontaneous generation. So as I said, this has an importance to keeping things pure. If we can prevent some of those bacteria or fungus cells from getting into our product, then it will be stable for much longer, or if not indefinitely. So, of course, the crux of these kinds of organisms and cells is that they are essentially invisible to us, at least with the naked eye. And so that's why we describe them as microorganisms. They are single-celled organisms, which means that they exist as just a single cell, as opposed to animals or plants that are made of many cells. A lot of fungi are also multicellular organisms. However, yeast are an exception to that, and yeast are also a single-celled organism. So yeast and bacteria, if you just had one cell, you couldn't really see it with your naked eye. And another aspect of microorganisms that I really want people to understand and that I want to emphasize is that yeast and bacteria are not the same thing, and they are not related to each other except very remotely by both being forms of life. And so they have substantially different types of cells, and they are not the same thing. So yeast are not bacteria. And so at this point we should get into a little bit of the detail of what kinds of cells make up these types of microorganisms and how they are so different. And so all cells, all living organisms, are divided into two main forms. They're called prokaryotic and eukaryotic. And so bacteria are prokaryotes, whereas yeast, which are a type of fungus, are eukaryotes. And the other organisms that you commonly recognize around, plants, animals, fungus, are all eukaryotes or eukaryotic organisms. There are a number of other microorganisms that are also eukaryotes. But bacteria are prokaryotes, and that name, prokaryote, is something that is describing, I'm going to get my pen here to emphasize some things, something that's describing a main attribute or difference between the two. So something that is a prokaryote means before the karyo, which is before the, literally it means the seed, but its meaning in this sense is the nucleus. Okay, so bacteria are before the nucleus, whereas eukaryotes means that eu means good or whole. They have a good nucleus. So that's the main, I guess you could say, really the main distinction between these types of cells is the presence of a nucleus in eukaryotic cells. Eukaryotes, though, also have a lot of other internal structures that we call organelles. They're much larger than prokaryotes by an order of about 10 to 50 times bigger. But that nucleus is the distinction in the name, and that's it is basically the main dividing line between the two. There are a number of other differences, however. And so we can see some diagrams of both a prokaryotic cell, or a bacterium, and in this case a eukaryotic cell, which is a yeast. And I'm going to come back to this picture a little bit later on when we're covering yeast biology and emphasize some of the parts of it. So at this point I just wanted to briefly do a comparison that bacteria and eukaryotes, so prokaryotes and eukaryotes, both have DNA in their cells, and that's what makes up their genome. So the difference between the two is that the prokaryotes, like bacteria, just have their DNA kind of arranged in an area of the cell where it's kind of held, but it is not set apart, it's not packaged inside a different structure. Whereas these eukaryotes have that nucleus, and the nucleus is what holds that DNA genome. And so this is a prominent or the main feature of them. Gets a little bit to get a little bit more into kind of the diversity of life and to understand this separation. We can kind of see things in a very big picture. If we look at a family tree of all kinds of life, just to demonstrate how actually far apart or how unrelated bacteria and yeast actually are, we can look at what's called the tree of life, and there's many ways that one can construct something that shows all the different forms of life and how they're somewhat related to each other. This one happens to be based on genetic sequences, and it's was really kind of a game-changing discovery or bit of work that went into utilizing genetic sequences, in this case of what we call ribosomal RNA genes. What are those? They're basically a part of the ribosome structures, but this RNA has a specific genetic sequence that codes for it, and by analyzing that and comparing it, since all forms of life have it, one can look at those differences and infer essentially how they are related to each other. So I won't go into it more than that, but just to highlight here how we have bacteria as well as another group of prokaryotes called the archaea. And these are then believed to be the most earliest evolved organisms, and you can see some diversity within them. But within the bacteria, we have one group called the gram-positives, and it's here that the lactic acid bacteria, such as lactobacillus and pediococcus, that you probably have heard about already and that we'll talk about some more, this is where they would be positioned. However, yeast are way over here in the fungi, and in fact the closest other types of organisms to yeast are actually animals, even more so than plants. So that kind of rounds out the general introduction to what is life and how things all kind of fit together.